Good morning, everyone. Let me share with you how we have used metabolomics to understand rice grain quality. Rice is life to billions of people, and the population of rice consuming countries continues to increase every year. By the way, this is not my child, but my daughter loves rice the same way that this child enjoys his rice meal. The rice production every year also increases. In the recent report published by Food and Agricultural Organization, the total production of rice paddy from 2018 to 2019 is around 759 million tons. 89.7% of it are produced from Asian countries, while 4.9% and 4.8% are from America and Africa, respectively. Clearly in this map, it shows that majority of the Asian countries produce more than 3.4 million tons of rice every year. And 2.9 kilocalories or 2,868 kilocalories of the energy that we need are from the cereals. 20% of it are from rice. There's a study shows that although the population in rice consuming countries increases, some of the consumers from developed countries started to diversify their meals and an observed decrease or decline in the rice consumption every year. However, for the Philippines and other developing countries, the increase in rice consumption increases as well every year, okay, as our population increases. This is a good illustration of how from 1980 to 2010, how much rice we have consumed and increased consumption every year from 92 kilogram to 119 kilogram every year. This is, by the way, for every Filipino's average yearly consumption. 119 kilogram is equivalent to 5 cups of rice. I'm not sure about you, but I'm trying my best to lessen the number of cups I eat every day to help the Philippine agriculture. Rice is also considered scarce. There is a challenge that we have to produce rice under fewer land and less labor. It's because mainly of industrialization and less people are engaging in agricultural activities. This is a good illustration where rice, wheat, and maize rice farms reduced significantly from the 1960s to the to year 2000, and significant decrease by half of the hectares of land dedicated for agricultural production. However, there is a good news. The Department of Agriculture made its press release in May 2020 that it says that we have enough rice supply adequate for the whole year, and I hope it will last until the 2021. And the Department of Agriculture is maintaining its um, goal of keeping a 2.5% growth every year, increasing as well the 2022 budget. Okay? I guess the rice is scarce is not real for the Philippines, so I should write here rice is enough at least in our country. Growing rice is a challenge. Cultivation of rice under suboptimal conditions are the challenges that the farmers as well as the rice breeding programs are currently facing. To produce 1 kilogram of rice, it says that we have to utilize 10,000 liters of water. That's a lot. But recent advances in breeding programs like marker assisted or molecular breeding were able to develop different types of rice that can adapt to different conditions, such as rice varieties that can tolerate submergence or flooding, mainly because of the presence of a quantitative trait loci or a gene responsible for flooding tolerance or submergence tolerance, as well as rice varieties that can withstand very minimal water, supply of water, mainly because of the drought genes. Some rice varieties are also being developed to cater for drastic conditions like high salinity and pest infestations. So if you look at it, yes, there is a challenge, but science is doing its part to continue to address to this moment, this big task. For rice chemists like me, it is our goal to provide information on how we can produce good quality rice, not only of amount, but also of quality. That is our goal. Majority of the rice breeding programs have rice grain quality laboratories that do the routine analysis for them. Among the properties that they analyze for breeding programs are the physical properties and the cooking properties. Physical properties are mainly focused on grain length, shape, and chalk. These are the characteristics that consumers um, easily see at the market. So definitely the physical properties command a higher value when you are dealing with prices in the rice market. The length of a grain refers to the dimension of the grain itself. Surprisingly, for us in the country, we are more often introduced to rice varieties that are long and slender. But actually, rice grains comes in different forms, from short, medium, long, and very long as its length, and bold, medium, and slender for its shape. By the way, shape is defined as the ratio of the length and the width of the grains, where we have bold, medium, and slender. Why are these shapes important, or why are these dimensions important? Mainly because of cultural preferences. Majority of the Southeast Asian countries prefer the long and slenders like this basmati rice, while for Eastern countries like Japan, they would prefer the short and bold type of rice. If you notice the sushi that we eat in Japanese restaurants, they are the cute grains that we see because of those short and bold shape. Chalk, on the other hand, is the opaque um, appearance that you see on the grains and the sperm. The chalkiness affects the overall look as well as the um, tendency for the grains to break when they are milled or polished. Of course, if you see broken grains, it commands lower value than those grains that are whole or intact. That is why when you see um, grains or a bag of rice that are full of broken grains, it is usually of cheaper price compared to those that are of whole grains. And chalk is playing an important role in that. Aside from the physical properties, we also looking at the cooking properties of rice, such as the amylose content, gelatinization temperature, and gel, gel consistency. So amylose is a linear polymer composed mainly of glucose units linked primarily by alpha 1,6 linkages. Um, if you say that there is no amylose or waxy, these are the waxy rices, or we popularly known in the Philippines as the malagkit rice. So waxy rices are also popular in Southeast Asian countries like Laos. Low, intermediate, and high are those rice varieties that have amylose content ranging from 0 to 20 percent. And very high means it has more of the amylose than the amylopectin. What does amylose mean? It means that it's the tendency of your, aside from the stickiness that we observe in our malagkit, it's also the tendency of the rice to retrograde after cooking. Okay, retrograde means namamahaw or tumitigas after cooking. Gel temperature, on the other hand, is the temperature at which the grain starts to cook. It is where the crystalline structure of your grain starts to melt and your rice starts to cook. Okay, so we have gel temperature ranges from low, intermediate, and high. Usually, this is from 55 degrees to 85 degrees Celsius. Okay. Did you know that lowering the gel temperature just by 1 degree Celsius, or just 1 minute, I mean, so assuming that there are millions of household cooking rice and reducing their cooking time by just 1 minute, it would mean 25,000 years of energy less every year that is conserved. Aroma, on the other hand, is one of my favorite topics. I guess you'll notice that the majority of the topics that I will be discussing in the coming slides will be dedicated to the aroma of rice. Aroma in rice grains are usually just done either to classify the rice as fragrant or non-fragrant. Usually you do it, you put 1 gram of the rice grain, soak it with potassium hydroxide for an hour, and then you sniff it. But of course, it takes some time for a trainer to identify whether um, the rice variety is fragrant or not. And of course, just like this, sniffing also would result to oversaturation in the detection of the fragrance of rice. Recently, routine analysis are now being done to monitor the 2-acetyl 1-pyrroline, or popularly known as 2AP by gas chromatography. 2AP is what 2AP is the compound responsible for the flower, the jasmine, and the pandan that we usually smell when the rice is cooked. Actually, for aromatic rice, if you visit the field, even if the rice is not cooked and it's still planted in the field, you can still smell this pandan flavor. That is how strong 2AP is. So these are the common quality traits that we monitor when we ensure that the rice varieties that are being developed in breeding programs are of good quality. With all of these traits, it's still surprising that the actual rice market is not represented properly. Okay? What do I mean by this? Um, for the past with for the past few years that I've been working with rice, I've been part of the International Network for Quality Rice. These are chemists, manufacturers of machines, and rice breeders from 31 countries, all working towards producing quality rice. And from this survey, we have identified three famous rice varieties in each country, in each of the member countries, and summarized the common traits, such as the physical and cooking properties. Consumers within a region or within a country, if you'll notice here, have quite a diverse rice preference. Amazingly, for India, you'll see here the diversity of the shape, the length and the shape of the rice grains that they prefer. The bottom or the southern part would prefer the short and the um, bold type, while the northern part would prefer the extra long and slender type. Similarly, in Southeast Asian countries, where Philippines is part of, the amylose content is also quite diverse. Waxy rice or zero amylose is preferred, as I mentioned from the previous slide, is preferred in Laos, while low to intermediates are preferred in Vietnam, and Cambodia, while Philippines as well as Thailand would prefer intermediate to high amylose content. Okay? If you summarize all of these traits, combine them in one design, combining the four properties of length, three properties of shape, four amylose classifications, three gel temp, and two aroma, will come out with 18 trait combinations. And these are the countries and their rice varieties belong to. Amazingly, with 18 trait combinations, we thought it is enough to define or describe a particular rice variety. But no, we were surprised that for the Philippines and Brazil, particularly IR64 and Brazil's BRS Primavera, they both belong to long grain, slender, intermediate amylose content, intermediate gel temperature. It is considered non-aromatic, but people would know the major differences between an IR64 and a BRS Primavera. With this, it is not surprising that we need additional tools to define rice grain quality. And this is where metabolomics comes in. As has been defined, metabolomics is a tool for comprehensively profiling small molecules. I love this illustration because it says the hierarchy from the DNA, from the genetics, up to the metabolites. And what is good about this illustration is the metabolome is what is dictating the phenotype or the characteristics or the traits that we are seeing, particularly in rice quality. So with this, let me share with you some of the applications that we have done using metabolomics to understand the volatile compounds using a single platform. Several properties such as elements, volatile compounds, polar and semipolar metabolites, as well as primary metabolites using multiple platforms. And finally, combine genomics and metabolomics as a multi-omics approach and be able to identify the volatile compounds as well as QTL or the quantitative trait loci mapping that will be valuable in molecular breeding programs. Let's do first the single approach in volatile analysis. In any breeding program, you always start with the right parents. I guess in any partnership or relationship or any endeavor, you always look for your complement. In this population or in this study, IR64 and APO or ERI132 or also known as NSIC RC9 for commercial code are developed, are being developed at this time, some time ago, to capture the good traits of each of the parents. IR64 is one of the elite rice varieties in breeding programs. It's because of its good grain quality traits. However, like any other elite rice varieties that were introduced, by the way, IR64 was only introduced in 1985, they are quite susceptible to pest infestations, to drastic environmental conditions. That is why the yield are not always favorable. However, IRRI 132 or ERI 132, or better known as APO, let's call it APO, are very resistant rice varieties. They can withstand low um, water, soil water or moisture, like in irrigation. So we can safely say that it's considered as a drought tolerant rice based on this graph where the yield of IR64 irrigated as well as the IR64 drought have significant difference. That's why we can consider IR64 as a susceptible, drought susceptible rice variety. Apo, on the other hand, with Apo irrigated and Apo drought, the yields have differences, but these are not significant, which means that um, Apo can withstand drought conditions or can resist low water supply. If we look at their grain quality traits, IR64 as well as Apo have similar gel consistency, gelatinization temperature, as well as the amylose content. So if you just look at the gel quality, the grain quality traits, you won't be able to distinguish IR64 and Apo. However, when you do sensory analysis, as we have done here, we have conducted and trained panelists to differentiate rice grains from Apo and IR64 grown in irrigated and drought conditions using several descriptors like taste descriptors like the sweet taste and the sour or silage taste, mouthfeel like the astringent and water-like metallic, and the mouth and other descriptors like aroma like corn, sweet aromatic, sour animal, and hay-like and musky. So how do we come out with this is because previous analysis by Champagne and her group already conducted a sensory analysis of Apo and IR64. So to lessen the uh, number of descriptors that the panelists will be considering, we only focus on these eight um, sensory descriptors. And true enough, when we see, when we compare the IR64 irrigated and drought grains, there are significant differences with the um, sensory results, you know, where majority of the astringent, water-like, sour, and sour are missing in irrigated IR64. However, for apple, whether it's in irrigated or drought conditions, you won't see much differences between the grains. By the way, the plus signs indicate the intensity of the descriptors as the panelists are describing them. When we do the metabolomics of the volatile compounds, we came out with, we were able to identify around 187 headspace volatile compounds. This is the principal component analysis of 12 biological replicates of APO and IR64. So the blue ones are the IR64 irrigated, the green one is IR64 grown in dry condition, while the red and the gray ones are the apple grown in irrigated and dry conditions. You will notice that IR64 grown in the irrigated significantly have a different volatile profile than those of the IR64 grains that were grown in dry conditions. But you won't see much differences between the volatile compounds that were emitted by the headspace of the rice grains grown both in irrigated and dry conditions of the apple rice variety. This clearly shows that the metabolite profiles that we identify can approximate or agrees with the results of our sensory analysis, which is quite interesting because conducting sensory analysis is quite tedious and it takes some time to be able to train panels expert for a particular sample and particular study. Another example of using metabolomics is using multiple platforms. Um, with the collaboration, we were fortunate enough to be part of the Metaphor project, metabolomics with partnership with different research institutions in Europe we are able to identify different properties of rice grains, such as multi-elements, using ICP-MS from University of Copenhagen in Denmark, volatile profiles of our rice grains based on the GCMS solid phase microextraction done by Wageningen University in the Netherlands. Um, Polar primary metabolites by GC, time of flight mass spectrometry done by Max Planck Institute in Germany. Semi-polar and polar metabolites as done by nuclear magnetic resonance in the laboratories in Rothamsted, and direct infusion mass spectrometry both in the negative and positive mode in also in Rothamsted. Okay, so with this um, different platforms, we were able to see the effect of different parameters like storage. What happens when a group of jasmine rice varieties are stored, um, and how are their volatile, since jasmine rice are very famous aromatic rice. Um, what is the effect of storage with its overall fragrance? This is the composition of all of this um, um, measurements, and came out with a principal component analysis that looks like this. Okay, for freshly stored or freshly harvested jasmines like RD6, KDML 105, or Kaudak Mali 105. This is the elite jasmine rice variety. RD33, as well as the Patam Tani 1, which is also the second best jasmine rice in Thailand. Clearly, their profiles are clustered together, which means they share almost similar characteristics, whether it's the volatiles, polar or semi-polar metabolites, as well as their elements. And as you increase the months of storage from 6 months to 12 months, you'll see a clear differentiation between the stored grains as well as the harvested jasmine rice varieties. With this, we can say that metabolite profiles indeed can identify whether rice is stored or freshly harvested. And we can use this information to um, see, determine the proper post-harvest treatments to ensure that aromatic rice like jasmine are kept in its finest quality. If we are to compare now two very famous rice varieties like jasmine and basmati. By the way, this picture was taken during one of the conference that were majority attended by members of the INQR, or the International Network for Quality Rice, as well as the Metaphor, who analyzed the rice varieties. There was a raise of hand or a survey of who prefers jasmine. So this is our good friend from Thailand, who proudly raised her hand to show her love for jasmine rice, while this is our another friend from Iran that would prefer basmati. So she's waiting her turn to raise her hand for basmati. By the way, I am the lady at the back also raising my hand in favor of jasmine over basmati. So if we compare these rice varieties based on their location grown or their origin, you'll see a significant um, clustering between the jasmine and the rice and the basmati. Here, you'll see the RD33, KDML 106, 105, RD6, as well as the PTT1, all clustering based on their volatile profile. Similarly, for their polar primary metabolites, as well as their semi-polar and polar metabolites, you'll see a cluster of jasmine in one side and another big cluster of the basmati rice varieties on the other side. By the way, the rice varieties of the basmati rice varieties are from members in from Iran and Pakistan, who gave us the different basmatis like basmati 370 um, and Supa basmati. And if you notice, there's a indication here that these are the fresh basmati. We clearly indicated that we want to compare all the fresh samples. No, it was difficult at first to find fresh rice, basmati rice, because the basmati is commonly stored for a year before they are sold in the market. So if we look at the basmati that are available in the market, they are usually characterized by off flavors or off scent. No? But they are stored, as I found out from our friends from Iran and Pakistan, as well as in India, that these rice varieties are stored because of the development of a better texture that are needed for their local cuisine. Okay, so they are not actually after the aroma, but rather more on the texture. Okay, so with this, we can say that yes, the metabolite profiles can actually differentiate rice varieties depending on their origin or their location. What is the practical application of this is we can identify whether rice basmati that is claiming to be from Iran can be identified based on selective aromatic compounds that are unique from basmati that are purely from Iran. Okay, so contaminations or adulterations can be monitored. This is one good application of this type of information. Similarly, IR60, no, not IR64, but rather the KDML 105 or the jasmine that is commonly sold in the market are also adulterated and contaminated with other non-aromatic um, rice. So it will be a very useful information if we'll be able to identify um, markers, volatile markers for this particular type of rice. Okay, so now we have shown that single and multi-platforms can differentiate rice varieties, whether it's um, their tolerance to drastic conditions or their locations or grow or yeah or their origin. Okay, another approach that we also want to look at is the multi-omics, no? Combining genomics as well as metabolomics. The genetic basis of rice aroma based on 2AP is already known. We already know that the betaine aldehyde dehydrogenase gene is responsible for the production of 2AP. That is why after the discovery of this gene, now there are three ways of identifying rice varieties, whether they are fragrant or not, in a rice breeding program. First is the sniffing, the second is the GC, and the third is through this molecular mapping approach. Okay, so how does it work? So we have in rice we have 12 chromosomes. On chromosome 8, in its exon 7, there is a deletion of um, information. This deletion results to a non-functional gene. So if the gene is functional, that means it's complete and there's no deletion, the betaine aldehyde dehydrogenase will produce GABA or gamma aminobutyric acid that is used when the rice plant is under stress. Okay? But if there is a deletion, especially on this region, there is a malfunction or non-performance of this gene, resulting to an aromatic rice or the production of 2-AP. This deletion is currently being developed as a screening tool and expand the capacity for early detection of fragrant rice varieties. Because with this method, you do not need to mature the plants, harvest the grains, and sniff or analyze it through the GC. With the DNA extracts, you can identify these alleles and be able to classify whether your rice at an early stage is fragrant or not. So from these publications, um, there are around 10 alleles that were identified, whether it's a deletion, insertion, or a change of SNP, single nucleotide polymorphism of G to T or C to T. So these alleles are now being used routinely to screen for early stages of rice varieties. It's really cool. There is a big advantage for this. However, this is like any other rapid test. You won't be able to quantify the level of 2-AP that the rice variety is producing. Unlike for gas chromatography, which is I am biased for, can quantify the amount of 2-AP in per every gram of grains produced. Okay, so we have done drought conditions in the Philippines. We have also analyzed using multi-platform metabolomics using rice varieties from Southeast Asia and South Asia, so from mainly Thailand, Iran, and Pakistan. Now we're moving towards South Pacific, mainly in Australia, where there is a breeding program to develop fragrant rice varieties using elite lines from Thailand, Laos, Cambodia, and use non-fragrant rice varieties that are of good quality, like IR64, Dungara. Fragrant rices also from Australia, like Kayima and Pandanwangi from Indonesia. So this is part of a breeding program, and we want, because of its unique um, um, collection of all of these combinations of parent rice varieties, we can actually understand better if there is a um, other genes that are responsible for 2-AP. Okay. Analysis of these compounds, analysis of these rice varieties and their progenies from the different populations resulted to this principal component analysis, where we have several compounds. Majority of the fragrant rice varieties are on top of the PCA, while at the bottom of the PCA are the non-fragrant lines. Interestingly, this is actually a biplot where you can see the rice varieties as well as the compounds or the volatile compounds that were analyzed from the headspace of the rice grains. So these are the triangles that you'll see in this PCA. What is interesting about this is this set of compounds. If you notice here, the triangle coded as 1 is actually 2-AP. And it's not alone in the PCA. It's clustered with 4 other nitrogen-containing compounds. 3 that we were able to identify, like pyrrole and 2-acetylpyrrole, but 2 that were needs further identification. So what we do with these 2 unknowns is analyze them using proton NMR. We synthesize 2-AP, and surprisingly, every time we synthesize 2-AP, there is another peak that comes out. And this could be the analyte 5, which is 6-methyl-5-oxo-2345 tetrahydropyridine, or probably it's better to just call it 6-M50-TP. Okay? And the analyte 4, we submitted it for further analysis using a 4D GC high resolution time of flight mass spectrometry, or a GC by GC HRT 4D. And from here, we were able to narrow down the identification of analyte 4 as 1-pyrroline. So all of these are nitrogen-containing compounds. And if we look at the genomics using genome-wide association analysis, surprisingly, these 2AP and the rest of these 5 and these 4 containing compounds lie in the same region or co-localized in a single QTL. This is marked with this yellow triangle here. And if you trace it with all of the populations that we have analyzed, let's trace this one. So all of the populations with 67, 71, 21, 36, and 25 progenies or lines are all positive with 2AP. While those that have negative 2AP, this QTL does not exist, which is coded in green color here. So exciting. So metabolomics have proven that these 5 nitrogen-containing compounds are similar, and also their region in the chromosome are also similar, which might lead us to, would they share the same biosynthetic pathway? So this is the published biosynthetic pathway of 2AP in 2008, wherein 2AP is produced from proline, ornithine, and then putrescine when it is converted to aminobutyraldehyde, and the BADH2 gene is functional. It will prefer the production of gamma aminobutyric acid, as I mentioned from the previous slide. However, if there's a deletion in the exon 7 in chromosome 8, the gene is non-functional, it's not working, so aminobutyraldehyde will form a cyclic compound, which is the delta pyrroline, and later on acetylate to form the 2AP that we are looking for. So with this mechanism, will there be another biosynthetic pathway or a modification of this biosynthetic pathway to understand how these 5 nitrogen-containing compounds are related to one another? And thus we propose the following mechanisms. We also believe that it's always from putrescine and aminobutanol. Okay, and we know that when it's there's a functional BADH2 gene, it will produce the GABA, but when it's not, it will produce the 2AP. When it forms a very unstable 6-amino 2-hexane dione, it will not stay in this compound, but rather it will immediately form a cyclic like 2AP, 2-acetyl-1-pyrroline, or the other compound like the 6-M5-OTP. Similarly, from 4-aminobutanol, you can also form 1-pyrroline, or you can also form it from pyrrole. Okay. And from 2AP, we can also produce 2-acetyl-pyrrole upon oxidation. So using metabolomics and genomics, we were able to propose another or modify a biosynthetic pathway for the production of 2AP and the other volatile compounds. And lastly, we know that rice aroma is not just about 2AP. We know that when we smell rice, it's not only the pandan smell that we can detect. So what we've done is move to a nearby country called Cambodia and use their world's finest rice variety called Pakarum 2 and couple it with an agronomically good rice like Tamar Creme or TMK and see how the metabolite profiles of or the profiles of their volatile compounds look like and see their genetic if there's also some genetic information about it. So again, similar to what we have done from the previous studies, we have analyzed it using the headspace of the rice grains and use a two-dimensional gas chromatography to efficiently separate the volatile compounds. Surprisingly, from this analysis, we have seen that there are significantly a lot of volatile compounds that are present in um, the rice grains, not only 2AP, although 2AP is the leading rice volatile compounds here with pandan described as pandan or the sweet flowered smell. Others are also present like pentanol, which is considered to be uh, with an other descriptor of green or grassy, heptane, which is quite sweet, and others would have some wine or nutty scent. If you also notice here, with the next to the other descriptors are the other threshold. Other thresholds in terms of the fragrance are quite important. Other threshold indicates the concentration that is um, needed for our humans to be able to detect. What is good about 2AP is it has a very low odor threshold of zero point 0.1 ppb, which means that even at a very small concentration, you'll be able to detect 2AP in rice grains. Unlike other compounds like this one that has 2,500 odor thresholds, that means it has to be present at that amount to be able to be sensed or be detected by our human sense. Okay, so using that population, uh, you're able to see a PCA that clearly divides the um, rice varieties according to the presence or absence of 2AP. And from here, we were able to see different volatile compounds like alcohols, aldehydes, carboxylic acids, esters, furans, hydrocarbons, ketones, and the nitrogen containing compounds. If we look at the cytoscape or a method of determining which of these compounds are related, you'll also see that 2AP as well as the other nitrogen containing compounds like pyrrole, pyrroline, and the 6M5 OTP are also correlated. If we look at the SNP genotyping using Illumina Infinium, using a 6,000 SNP bid sheet, and Tassel and QGene for QTL analysis, we are able to construct the following Manhattan plots where majority of our aldehydes and with an LOD of more than 3, which is quite good, are detected here. So here are pentanol, hexanol, heptanol, octanol, nonanol, and these are quite important in terms of their contributions to the overall aroma. And these are quite useful if we will consider a particular rice variety to be developed and that would have high amounts of this aldehyde. So in summary, single and multiple platform metabolomics can reveal unique metabolite profiles of rice, whether it's based on their origin or location or they're grown in different environmental conditions. And we have proven that metabolomics combined with genomics would be a powerful tool to identify metabolite QTLs that are important in marker-assisted breeding programs. And also, it helped us formulate new hypotheses for aroma production or biosynthesis. And we have proven that using multi-omics approach, it will uh, surely pave way to better understand rice grain quality. With this, I would like to thank the following: the International Rice Research Institute, Plant Research International, and the Plant Physiology Department of Wageningen University, the School for Agriculture and Food Science of University in Queensland, and Monsanto Bichel Borlag for my PhD, and Agents of Change Industry Transformation Training Center of the Australian Government, Australian Research Council for my postdoc fellowship. Again, thank you for listening.
Ayun sa